or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a Ph.D. in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content.
Nick.
Can you start off by just telling everyone a little bit about who you are and what your lab studies?
Yeah, so my name is Marcelo Dietrich. I'm an associate professor here at the Yale School of Medicine, and my lab is particularly interested in the dynamic change in neurocircuit that happens throughout development. So from infancy to adolescence to adulthood, how neurocircuits are changing to regulate distinct behaviors and physiological needs. So that's what we are studying.
And on your lab website, you have a nice lab website. You have this video and a quote from a man named Harry Harlow. Can you talk a little bit about what that quote means and who that was?
Yeah. So yes, Harry Harlow. The quote is that if we have learned anything about the studies of monkeys, is that we have to learn how to love before we learn how to live. And it's a long story. I will try to make it short. So Harry Harlow was a primatologist, a behavior scientist who made really pioneer studies in the 50s, 60s. And we can talk more in details if you want, but basically what he was trying to test is what are the factors in a mother that are relevant for a baby. So he took orphan monkeys. He would actually fly these monkeys from India. He was a pioneer in studies rhesus monkeys. And then he would raise these monkeys with two types of wired mothers. One wired mother that had a bottle of milk, and one wired mother that had a clot. And so the clot would provide some kind of tactile experience. And the idea was to test the hypothesis that whether the monkeys will like better a milk or tactile experience. And those studies were really mind-blowing back in the day because the monkeys would spend like 23 hours and a half of the day wrapped around the clot mother. So they would overwhelmingly choose the clot mother.
So the idea was, well, obviously the baby needs to eat and the mother is the source of food. Obviously, babies love to be embraced by their mother and hear the mother's voice and all these things. But when you put the food, the milk, against the physical touch sensations of the mother, you're saying that the monkeys would spend almost all of their time getting that physical touch. They really seem to prefer that.
Yeah, correct. And I think those findings need to be put in the context of the time of the 50s and 60s, and we can also try to go through that. But I think the most fundamental experiment of Harlow is the following. We have all been in a playground with kids, and we see that the kids usually look for the caretaker, for the figure that's around, to see if the caretaking figure is around, and then they play. So if the caretaker disappears, the kids flip out. So Harlow had that intuition, so he did the same thing. He made a playground for baby monkeys, and he put the baby monkeys to play in the playground. But then he put the wire mother with a bottle in one playground, or he put the wire mother with a clot. So when Harlow put the babies in this playground, and there was the wire mother with a bottle of milk, the babies will not play. They will freeze. They will be in tremendous distress. So that mother provide no comfort for the baby, despite the fact it provide milk. When he then switched the mother and put the clot mother, these babies monkeys will go hug the clot mother, and they will be playing around, and then often they will look back, see that the mother figure was there, the attachment figure was there, and they will continue playing. So that was really, I think, the key experiment showing is not only a preference, but this artificial inanimate mothers have actually the capacity to buffer the distress of the infant and allow the infant to explore and play and learn about the world. And so, yeah, that's a experiment that I like very much.
Hmm. Yeah, and it starts to tell us that I guess there's multiple things in an infant's environment that are tied to its mother that are of interest that the baby needs. It needs food, it needs safety, and to have a protector around. It needs that physical touch, but all of those things are not weighted equally to the infant. That some of them are having sort of a stronger effect on the baby.
Yes, certainly. And yeah, Harlow tests that, and I think there's a lot to talk about Harry Harlow, but it's probably one of the saddest experiments. He did a few as he progressed in his career. He got a little out of his mind and was heavily criticized for
Hmm.
his, the ethics of his experiments. And one experiment that he did was to cool down the mother.
Hmm.
So he cooled down 2 degrees cooler than the room temperature and ask whether the baby would attach and would get milk from the mother.
And this is the artificial mother.
Artificial, yeah,
Yeah.
artificial. And he did, I believe, I might be incorrect on this one, so we'll have to double check. And but I think he also anesthetized the mother and cool down. So if he cools down the mother 2 degrees under room temperature, the infant monkeys will never get close to them, will not get even milk, and they will die. So actually, the title of the paper is Psychological Death in an Infant Monkey.
Whoa.
So, and yeah, very sad. So basically, in Harlow's experiment, he puts temperature as number one
Hmm.
of the sensory importance of the mother, and then comes touch, tactile experiences, and then rocking. The movement is important.
Mm.
Milk is actually the last of the series. It's not that it's unimportant, but nursing
Hmm.
is the lowest in importance in his experiments.
Hmm. Yeah,
Yeah.
and it's potentially counterintuitive, right? You might think, well, the baby needs to eat to survive. The mother's the sole source of food. It's a little bit surprising. It's not surprising that that the tactile sensation and the warmth are all important, but it is perhaps surprising that they're even more important than the food.
Yes, certainly. And that's why I like that quote that's on my website, because also at least I think if we talk to child psychologists or people that are in that field, they will not find surprising, at least now. But for us, when we start studying this, was also surprising. And I think the concept that's always on my mind it's the ecological context in which these features evolved.
Mm-hmm.
So, and here we are talking about mammalian evolution. We have about 6,500 mammals on Earth, and all mammals necessitate a mother, a caretaker, to obtain this early nutrition and early stimulation. So the way I think, and I can explain that those Harlow's findings in an evolutionary context is that the primary importance in the beginning might be the milk, but after millions of years evolution, the mother always predicts milk, hence milk
Mm-hmm.
becomes irrelevant from the perspective of evolution. What is more relevant is the other stimulus. So the proximal effect, as we say, it's actually touch and other stimulation, but the ultimate, you definitely need milk. But the correlation is always 100% in
Yes.
nature.
Yes.
You never get a
Yeah.
mother the wild. And if you do have a mother, usually that provides no nutrition, the infant will die. So there's no evolutionary
Yeah. Yeah.
adaptive selection for that.
It's just like applying reinforcement learning within one animal's experience across millions of years of evolution. The most efficient thing to do is to learn the first thing that predicts the outcome you care about. And in this case, right, the mother is always around to provide the milk, so it does make sense when you think about it in that way from a reinforcement learning perspective, that evolution would sort of bake that kind of tendency in.
No, I agree. There's another animal that I like to talk about because I think it's just fascinating. So as I say, we have about 6,500 mammals on Earth. And the one that we know that has the shortest lactation period is one seal that lives in the Arctic. It's the so this seal, the infant only the neonate only stays with the mother for 4 days.
Hmm.
So within these 4 days, we'll gain like 200 kilos or so. And so we'll take an immense amount of milk. And then the baby will leave, but the baby cannot hunt on their own for another month. So they survive, they fast for another month, only eating ice as a form of hydration. So
Hmm.
actually, the physiological systems to eat are not ready, but ecologically, the baby cannot stay surrounded by the mother because there's a risk of predation. But even in that extreme evolutionary context, ecological context, the attachment, the relationship of the baby with the mother didn't disappear. It did shrink to 4 days,
Hmm.
but it did not disappear. Maybe we need a few more millions of years to find mammals that in which that bond will be broken. But that's one of the extremes.
Yeah. So that just gives a sense for the strength of this bond and the importance of that.
Exactly. That's
Yeah.
is very is basically a constitutive part of being a mammal.
Hmm. And one of the things we're going to get into here is how hunger and feeding work and how some of the circuits in the brain develop that regulate that. But obviously, just on an intuitive sort of experiential level, obviously an infant has feeding behavior that's very different from an adolescent or an adult. As adults, we eat so many different types of foods. We have to learn what we like. We get to test out all different sorts of tastes and see how we react to it. But for a newborn infant, right, it's sort of all baked in. There's going to be one food source for some period of time. It's just the milk. The baby doesn't need to learn about the milk really. The suckling reflex is going to be there right from the start. And as you said, the mother's obviously there when it comes to mammals. That's just part of being a mammal. Can you talk a little bit about how sort of hunger and food and nutritional needs change from infancy to adulthood and sort of general ways that cut across all mammals? Obviously the milk has a certain nutritional composition. It's presumably tuned to the baby's needs early in life, and those are going to shift over time. But what are some of the ways, the major ways, and most are all mammals, that sort of the food needs and the hunger needs of an individual change from infancy onward?
Yeah, it's a fantastic question. There's so much to unpack there. So many things come to my mind. Maybe we break down in piece and you can reflect on what I say.
Hmm.
Since we are talking about different animals, different mammals, and examples of that, I will continue giving these examples because I think this one is mind-blowing too. So marsupials, some marsupials, so mammals that will have a pouch and the embryo will actually attach to the nipple within the pouch
Mm.
and then will grow within the pouch. Some marsupials will have multiple layers of nipples. So, for example, we'll have the vagina will have three chambers. So the first embryo climbs from the chamber and attaches to the first layer of nipples. And then once it grows, it climbs to the second layer of nipples, and then the embryo from the other chamber will come to the first layer. So they are synchronized in the climbing the layers, and each layer will have a different milk, which will be important for the growth and nutrition and the immune system, immune protection
Hmm.
of the growing infant as it grows in the external
Hmm.
pouch of the marsupial mother.
So if I'm hearing you
Kangaroo.
right, so in marsupials, the embryo is developing and it's always in the pouch, but there's literally different sections of the pouch, and the embryo is kind of climbing through each section as it grows. And as it grows, but while it's still, in the pouch, in the womb, I guess, um, it's attaching to different nipples, and the composition of the milk in each nipple is actually different.
exactly. So
Wow.
I give that example to illustrate this remarkable features. It's the same mother, different nipples, each nipple with a different milk composition. So what we do know is that milk composition changes throughout lactation in animals and primates and humans,
Hmm.
and that matches the nutritional and also the other needs. For example, milk will have many antibodies that will be important for immune protection of the infant early on. For example, IgA, that's a mucosal antibody, will be very high early on, and then we will start
Hmm.
fade as lactation goes on, as also the infants start produce their own levels of antibodies. So there are all these features that are matching the physiology of the infant. So if so, that's talking about that. So um, maybe you.
Yeah, so basically, I mean, obviously, the nutritional needs of a baby are different than an adult. But what you're saying is like even within the preterm period, even within early infancy, the nutritional needs are changing. So the composition of milk, from when a baby's born on day one will be different, just a few weeks or months later. And that's all tuned to the baby's needs. There's the nutritional needs. There's the sort of immunological effect here. Before the baby can produce its own antibodies, it's literally ingesting them from the mother. And so all of these things sound like they're sort of beautifully orchestrated to perfectly match what the baby needs each step of the way.
Yes, certainly. And so, yeah, one, and then the question you ask also the question about regulation, right? There's a regulation of feeding in adults. Well, that we know quite a bit about that, and how is the ingestion regulated in an infant
Mm-hmm.
or in a neonate? We don't know fully. That's the real, I think the most accurate answer. And there are many theories of whether breastfeeding and intake of solid food, for example, utilize same neurocircuits, same brain processes or parallel brain processes and things like that. So we still don't know the answer to that. But, however, we know a few things, mostly from rodent models, I would say, rats mostly, some work in mice, that very early in development, right after birth, they're, the neonates don't seem to have a mechanism to stop ingestion
Hmm.
when they are ingesting like breast milk.
Hmm.
In other words, if you take a infant rat and the infant attached to the nipple of the mother, but instead of getting milk from the nipple, you pump milk in the mouth,
Hmm.
this baby will ingest constantly until almost literally exploding.
Oh wow.
Will only stop when the stomach is in huge, 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 huge.
So
So,
it sounds like that in an infant rodent at least, the satiety mechanisms that are developed in an adult are not there yet.
yeah. So when you put that in perspective of the relationship of what is happening in breastfeeding, it makes sense from the following perspective. The regulation of the intake is actually done by the milk ejection reflex.
Hmm.
And the milk ejection reflex is not constant. In fact, when the baby is attached to the nipple, is stimulating the sensory neurons in the nipple. In the rat, people have done that experiment. The ejection of milk from that stimulation is going to be a random event
Really?
with, within a within a certain distribution of time.
Yeah.
Let's
Yeah.
say within 30 seconds to 3 minutes or 4 minutes. I don't remember exactly the data. When the milk is going to be ejected is a random
Hmm.
probability. And I think that's really fascinating because you remove the need for the infant to have a regulatory process.
Yeah.
Again, the same evolutionary process. There's no need for regulating because there will not never be enough. And whatever comes.
Hmm.
It comes with the timing that's definitely sufficient for absorption, opening the stomach, going to the lower gut, and getting more milk, more milk.
Hmm.
So the
Hmm.
mother regulates that process. Again, you give to a third party the regulation of the self.
Yeah. So
So
so when you see an infant mammal suckling milk from its mother, it's not actually getting it's not like if you and I were to have a cup of milk with a straw in it and we can constantly suck it out at a constant rate. That's not what it's like. The milk is sort of coming out haphazardly, and this is actually serving an adaptive function. It's preventing the baby from ingesting too much, essentially.
exactly. And that's one part. And the other adaptive function is when the baby is there in the breast, in the nipple, many other things are happening beyond the ingestion of milk, much like the experiments of Harlow. So in that moment, for example, when you record the autonomic nervous system of an infant,
Mm-hmm.
the autonomic nervous system, the sympathetic tone of a baby is much more regulated, is much calmer when
Mm.
it's attached with the mother.
So the baby feels safe, the stress levels go down,
And
the baby's presumably not anxious or anything.
in that context, the baby learns.
Mm.
In that context, the baby, so it's calm, stress response is low. The baby learns about the environment.
Yep.
Very early days, we learn odor associations. We start learning sound associations. And then when
Hmm.
vision start to get better in the human baby, we start learning facial
Yeah,
expression,
yeah.
all in
So
that context.
yeah, and this would imply that I mean there's a lot you could unpack here and there's a lot of things you could think about in terms of, modern human caregiving for infants and stuff. So, just ask you one thing here. You could imagine two human babies. One of them is getting breast milk directly from the mother, the natural way, and doing things the natural way. Another one, you could imagine giving an infant formula from a bottle, same nutrition value, same calories, and everything. The implication here is that baby would actually have some learning deficits, maybe because they're not going to have this change in their sympathetic nervous system that's going to affect their stress levels, and that's going to have knock-on effects for their ability to learn faces and to become familiar with their environment.
Yeah, it's absolutely it's a generalization, but I think is fairly correct. And I think sadly, we learned quite a bit about that process from, I think your audience is familiar with that, probably with the Romanian orphanage. So in the late 80s, with the fall of the Berlin Wall in Romania, Romania in the 80s was in an economical crisis, so need more babies. So there was a governmental communist incentive to have more babies, so families will have a lot of babies. And there was a selection process, so babies that were kind of misfit were put like in these warehouses. So they receive nutrition, but really no stimulation, no caretaking.
Hmm.
And so when scientists got there after the fall of the dictatorship, they found there's hundreds of thousands of babies. So there was nothing that could be done. There were no families to adopt all those babies. So they run the only clinical trials that we probably will ever be able to run, which was selecting some kids and give to foster
Mm-hmm.
caretakers. And what we learned from that is that if you do take infants during a sensitive period, roughly the first 2 years of life, and you provide that care, the comfort, that tactile stimulation, they will develop quite well. But
Mm-hmm.
if you deprive them, so if you only give the bottle as you suggest, then these infants will develop with very severe neuropsychiatric symptoms and social deficits, impairment capacity to socialize.
Yeah.
So yeah,
Wow. Yeah. Yeah. So that early period development is super important, and it's not just the sort of raw physical needs of the body in the form of getting food and hydration and stuff. It's all of this sort of these sensory and social effects that put the nervous system in a state that actually just enables learning generally.
exactly.
If you don't have that, you're going to have all sorts of problems.
Yeah, yeah, absolutely. That's a yeah.
And so you've you've done studies in young rodents that have to do with disentangling how the feeding system develops and things like that. Before we get into some of that work that you've done, I want to give people a general sense for how the sort of we'll just call the hunger circuitry in the brain works in an adult first. So I'll let you sort of take it where you will, but the hypothalamus is going to be a very important brain region here, and there are specific neurons in the hypothalamus that are really cool and really important here that drive hunger and satiety. Can you give us a basic understanding for what some of the key circuitry is that controls hunger in mammals, in adult mammals?
Yeah, absolutely. And yeah, I will certainly focus on the hypothalamus because I think it will make sense for our discussions later on. So in the actually in the most basal part of the brain, so above our palate, our soft palate, there will be our pituitary, the gland that regulate many of the hormones like HPA, stress response. And right above that, there's a layer of thin skull and then the hypothalamus. So the
Mm.
hypothalamus is in the very base of the brain. And the nucleus, the part of the brain that's the most basal is called the arcuate nucleus. In the humans and primates, the infundibular nucleus. And in this region, there are some neurons that are important in the regulation of feeding and metabolism. We start to suspect this more than 100 years ago because that region, the pituitary gland, is commonly affected by tumors. So the tumors
Mm.
go, and because the hypothalamus is right there, it pressures the hypothalamus, so it start causing some people have symptoms in energy metabolism, obesity, or things like that. So in the 19th century, people start to suspect about the role of the hypothalamus in feeding.
Hmm.
So fast forward 100 years, we know there are two populations of neurons there. One is called HRP. HRP comes from a gut-related peptide, a gut-related protein, which is a gene, the HRP gene that produces this peptide by these neurons. And when these neurons are activated, or when the animal is deprived of food, these neurons will be activated and they will promote feeding and many of the metabolic responses to fasting or to food deprivation. Next to these neurons, there's a second population that's called POMC neurons. They produce pro-opiomelanocortin, and the pro-opiomelanocortin is a large molecule, a large protein that can be chopped in several small peptides. And some of these peptides have strong satiety effects like MSH, alpha MSH, gamma MSH, and also beta endorphin, which is an opioid. So the POMC neurons are thought to be the counter arm of the HRP neurons, having a little more delayed effect, but when they are active, they promote satiety. And HRP neurons, when they are active, they promote hunger. That's an overly simplification, but I think it's a
Yeah.
fair one.
So a bit of a simplification, but in essence, at the bottom of the brain, in what's often referred to as the arcuate nucleus of the hypothalamus, there's two neurons, and basically, if you turn on one type of neuron, the HRP neuron, animals look like they're very hungry, they will eat a lot, etc. And if you turn on the other POMC neurons, you basically get a counteracting effect.
Exactly.
And so naturally, you would think, what do we know about these neurons in terms of their activity? If you've got sort of a hunger neuron type and a satiety neuron type, you would imagine that turning on, say, the HRP neurons drives feeding behavior, that when animals are hungry and they're looking for food, those neurons might be very active, et cetera, et cetera. Is that essentially how they work?
Yeah, it's yes, that's uh so in the early 2000s it was shown that the HRP neurons will respond in that predicted manner. You food deprive an animal, you have increased activity of HRP neurons. You feed the animal back, and the activity will decrease. The techniques used back then were markers that have a very slow dynamics, which
Mm-hmm.
means matter of hours
Mm-hmm.
to find those activity changes. And in 2015, using new technologies, basically three labs almost simultaneously, Scott Sturson at Janelia Farms, Mark Enderman at Harvard, and Zachary Knight at UCSF, uh, using three distinct techniques, they were able to record the activity of these neurons in the live mouse. And they actually show that once the animal actually sees the food or perceives that the food is available in the environment, the activity of these neurons will suppress, will decrease, in fact, in anticipation of eating.
Hmm.
After eating, there's a further suppression that comes from gut signals, but that anticipatory drop was something that was somewhat unexpected if you think like only we call homeostat, like if you're changing your temperature in the house and
Hmm.
but like 72, and so the thermostat will only switch on and off at that point. But here you're saying that as it's reaching, it's predicting
Yes,
a
yes.
future, so it's actually switching anticipation to
Yeah. So
uh,
if yeah, you're saying if these neurons were merely regulating hunger in a purely like homeostatic fashion, they would be pretty simple. They wouldn't start to change their firing until an animal eats the food and the calories get into the body and they're actually like ingested. But and that does happen to some extent, but what you're saying is when an animal even hungry animal even sees food, these neurons are immediately responding, and so they're doing something more sophisticated and complex that has to do with understanding that food is about to come even though you haven't actually ingested it yet.
yeah, absolutely. I think that comes back again to the complexity of the brain and of mammalian systems in general, that we are not dealing with engineer like machine
Mm-hmm.
that turns on and off, and it's a much more complex system in which one of the main advantages of this complexity is really the capability to predict what's going to happen at different time scales, seconds, minutes, hours, days, and
Mm-hmm. Mm-hmm.
so and that really gives a lot of adaptive value to the system because you
Yeah,
can.
and oftentimes the caricature that people have of the hypothalamus is, it's deep in the brain. It's part of our so-called reptilian brain. It's sort of a stupid area of the brain compared to maybe the cerebral cortex or something like that. What does this start to say about, the sophistication of the hypothalamus and how much that's been maybe underestimated over time? And to what extent do these circuits actually tie into learning and memory systems that we think of as being higher up in the brain normally?
Wow, that's a very good question. I'm trying to think how to answer this question without diverging too much because I think there's a lot to unpack in this HRP. Uh, um, uh, I think one way that I can answer, I think we still don't know fully. That's,
Mm-hmm.
I think, is a fair answer of how these hypothalamic circuits, how they interact with higher order learning circuits.
Mm-hmm.
Um, Now, let's say an animal sees a sound. That sound has no predictive value. It's the first time the animal sees a sound.
Yeah.
The AGP neurons will not do anything. You record the activity, they're not going to do anything.
They have no relevance to food and
No
satiety or
relevance.
anything. Yeah.
Then you pair that sound with food. You give the food. Uh, so every time the sound rings, there will be food that will come with the right after. So then when you give the sound, the AGP neurons are going to start to respond. So there's definitely a learn component. So the animal is learning that the sound now is a predictive cue of food, and that cue is actually coming from higher brain orders, sensory systems, all the way to the HRP. We know a little bit about the circuits that engage that cue evoked suppression of the HRP activity. However, we don't know fully yet how this memory,
Yeah.
how the memory of that context is actually going all the way. And I think that's a very important question. And I think there
Mm-hmm.
are many laboratories right now trying to actually figure that out.
Yeah. Yeah. But I guess,
Uh,
at a high level, the circuit is not purely responding to hard-coded, instinctive, food-related things. It is actually receiving information that's learned through experience.
100%. Yeah, 100%.
And so what happens, just to give people a little bit more sense of how important these neurons are, let's say you take an adult animal, and I know that there's ways to do this, and we don't need to go into the technical methodological details, but scientists have the ability to, say, destroy AGRP neurons in an adult, and people have done this. What happens if you destroy these neurons in an adult animal?
So it's another good question. It's The findings have been a little controversial in the last couple of years, but I would tell you the see, originally in 2005, the laboratory of Rachel Palmiter, they created a transgenic mouse line that allowed eliminating the AGP neurons in the brain acutely. So expressing the receptor for diphtheria toxin. So when you give the diphtheria toxin, will bind to the receptors in the AGP neurons only and kill these neurons. When he did that, the animals will starve to death
Hmm.
within a week. And so that was thought to be the case until recently. Now, when he did the same procedure in a 2, 3, 4, 5 days old animal, so a neonate, nothing happened.
Hmm.
They didn't die. So originally in his paper was a paper published in Science. I remember vividly the day the paper came out, and so it was thought the age of neurons were essential for feeding in the adults, but not in the neonates. Now, that concept has been put a little bit in check recently with another study that used several of these techniques and said that maybe the effect of the ablation had some indirect effects, was not exactly by killing the AGP
I
neurons,
see.
but
So it
some
wasn't
other.
fully, maybe not a fully clean experiment. It was doing something other than ablating the AGRP
Yeah,
neurons.
maybe. And I think it's still very controversial because many people have replicated those findings, and
Yeah.
I think these techniques all have their caveat. Now, in general, if you don't have AGP neurons or inhibit the AGP neurons, uh, the animals will at least have a deficit.
Yeah.
In
They eat less.
They will have a deficit in eating. If you food deprive them and give food, they will have less of that
Mhm. Mhm.
effect. So in general, that's the
Yeah. And I guess in any case, technical issues aside, one of the key results there was that you see a big difference in the type of effect you get in babies versus adults.
absolutely. And that always stuck to me in my brain, and it was one of was a key finding uh, to stimulate us many years later. So I started my lab in 2014, and I was interested in that question, though, what regulates the intake of breast milk, of
Mhm.
calories early
Yeah.
on,
Yeah.
if you can kill the AGP neurons and nothing happen.
Yeah. And I
Is
mean,
that
immediately I'm thinking of what you told us before, where the baby, there's this mechanism in place in the mother to make sure that not too much milk comes out, because the baby actually doesn't have the ability to self-regulate its intake, and so that would imply that the maturation of certain parts of the brain that have to do with feeding and satiety takes time and has not developed in the early infant.
Yeah, absolutely. Yeah, this concept, it's much clearer to me, and I think to us in general today than it was 10 years ago.
Mhm.
Uh, but that was somewhat what we tried to test. Basically, we asked the question whether these AGP neurons that promote hunger would regulate the intake of breast milk or would have any importance for the ingestive behavior of infant that's still in the food dependency on the mother. So the mother is the regulator. And so we did those experiments right when I start the lab, trying to play with the neurons, activating the AGP neurons, inhibiting the AGP neurons. And it was quite striking that we would find no change in ingestion of breast milk, for example. If we, doesn't matter what we did with the neurons. But when we would play with the neurons in the babies, for example, in an infant mouse that was alone, we could make these babies cry more or cry less.
Hmm.
So the infant mice vocalize in the ultrasonic range. And then, so we could have really big effects on the amount of this cry behavior or the amount of ultrasounds that the babies will emit when they were alone.
Hmm.
And we thought that that was quite interesting. And those ultrasounds are known to function as a cry, which means if the mother listens, the mother comes, it
Yeah,
will be
yeah.
attractive to the mother.
Hmm. So,
And
so just to unpack a couple things here before you continue, if you take an adult mouse
yeah,
and you turn on the HRP neurons, but there's no food around, they will start looking for food. They'll sniff the air, they'll dig through their bedding, they'll search around for food, and it looks like they're trying to find food. If those neurons are active and there's food around, they'll basically stop what they're doing and eat it. But what you're saying is when you do this in an infant, what it starts doing is basically calling for its mother.
exactly. They call for their mother, and then we did the sequence was more or less. I'm telling the sequence that we did the experiment. So then we look for if these neurons were active when the infant is alone, crying for the mother. And actually, they get very activated. They are very active, as much as one would predict after depriving an adult from food for like a day.
Hmm.
So we were very surprised. You separate the baby from the mother for half an hour and a half, and the activity is very high. Again, we use these techniques that have no temporal resolution. And so the neurons were active when the infant is alone, the AGP neurons, and when you inhibit these neurons, they cry less. When you overactivate them, they cry more, and
Mm-hmm.
that changes the mother behavior. And then we took some of these techniques that I mentioned that these labs used in 2015, and then we applied them to record the infant mouse, which is quite tricky because the infant mice are very small and they grow from one day to the next. But yeah, I also remember those experiments, and basically it was quite striking when we are recording the neurons and the infants are with the mother, with the siblings in the nest. The neurons are quiet. As soon as we take everybody out and they are alone, the activity of the AGP start firing immediately within seconds. So it's a very rapid effect.
Mm-hmm.
And as soon as we put any social partner back, be the siblings or the mother, the activity is suppressed to what it was before separation within 30 seconds.
Mm-hmm.
It's a very rapid alarm system, like turn on, turn off.
Mm-hmm.
That's very different from what we understood from how the activity of these neurons is modulated in the adult, because in the adult, the activity is slowly increases as the animal is deprived of food.
Yeah, yeah. It's a slow signal that's tied to basically how hungry the animal is.
Yeah. And here is within seconds.
Hmm.
So, and what was uh, interesting, the experiment that we did, very much inspired by Harlow, was separate a baby mouse, activity of the AGP neurons is high, and then put it back different
Mm-hmm.
kinds of foster mothers
Ah.
and see what is important for suppressing the activity
Mm-hmm.
of this.
And I guess naturally, to anticipate some of this, naturally knowing what we knew about AGRP neurons and the relationship to hunger, these are, this, people call them the hunger neurons, basically. You might predict, I might have predicted, I think, that if you took the baby away from its mother, the mother predicts food for the baby very reliably. So the AGRP neurons become more active. And if you then give it milk without the mother present, the neurons might get silent again because the AGRP neurons are supposed to be all about hunger.
Exactly. So we did that, the exact experiment that you said. So we pump milk back. Basically, was an experiment that we have to take mother's milk. So we pump milk from the mother and give the milk back for the pup. So the
Hmm.
pup will ingest their own milk,
Yep. So
their,
they're getting their natural mother's
their,
milk.
their natural mother's milk. So we did that. And we also did a cow whole milk from cow to have a different source of calories to control for that. And milk didn't suppress the activity of the neurons. So the
Hmm.
babies, we filled their stomach while they were alone, and it did nothing to the activity of the neurons. And then
Hmm.
we were puzzled because it broke our
Yeah.
expectations.
So these neurons
So
remain active. The so-called quote unquote hunger neurons remain active, even though the animal's not hungry anymore because you just gave them all the milk that their stomachs could hold.
exactly, it was around that point that the student that was the grad student that was working in my lab, his name is also Marcelo Zimmer, so we called him M2. And we were puzzled by that data. And then reading Harlow and all those studies, we say, how can we test that? It was then that we did the experiment that I was going to tell you before. We isolate the infant and then we gave different foster mothers. So instead of pumping milk, we provide different forms of caretaking behavior.
Mm-hmm.
And we thought first we provide a foster mother that the mice, like females, when they are not lactating, the nipples are not protruded, so the mice cannot attach to it.
Mm.
They don't have nipples that the baby mouse can attach. So, but if you take a grandmother type of thing, they will have no nipples, but they are caretakers, so they
Mm-hmm.
really
So
care
they
for
still
those.
feel the warmth of the mother, the feelings of her fur, the tactile sensations, but there's no nipple to attach to.
They will lick, they will groom, they will do everything that a mother will do, but there is no attachment to the nipple. So one type of foster caretaker was the grandma, we call the grandmother. The other was a female. We know from our own experience with infants, but also from the animal research and the animal world, that what's called the pacifier effect. An infant that's alone is in a distress, you give a pacifier, you stimulate them out, and that calms down the baby. Uh, I often give that example uh, for bat orphanages. In bat orphanages, one great way to raise bats that are orphans is actually to give a pacifier. They grow better.
Hmm.
So we thought that we need to give a pacifier to see if a pacifier will pacify the AGP. So one foster caretaker was a female mouse that had nipples. So now the babies could attach, but the nipple produced no milk. We experimentally generate that kind of female mouse. And the third type of foster caretaker was another mother, a different caretaker that had nipples and had milk. And then we ask which of the mothers will cause the largest inhibition. Against the prediction being that if these neurons are hunger neurons, the presence of milk should suppress when it comes from the natural environment, the breast.
Mm-hmm.
Maybe when we pump, we had no effect because it was artificial.
Mm-hmm.
And the results were striking. If we give a caretaker, a grandmother, no oral stimulations, just caring, we suppress the activity of
Hmm.
the EGP, let's say 30%.
Mm-hmm.
If that foster caretaker has nipples, therefore oral stimulation, we suppress, let's say 80%.
Mm.
Now, if the foster caretaker if has nipples and milk, it still suppress 80%. The
Hmm.
presence of milk has no effect
Hmm.
in further suppressing the AGP neurons. And that was really remarkable
Hmm.
at that time because you say, wow, it's really it's that concept that we discussed before. It seems that the milk is insignificant to modulate the activity of these hunger neurons during the sensitive period of life in which caretaking and nutrition is coming from the mother, is
Yeah.
being regulated by the mother.
Yeah. Yeah. So, I mean, it is sort of gets to this idea that these neurons, they're obviously about hunger, but it's less about, it's not completely about the presence or absence of calories. It's about the presence or absence of sensory stimuli that will predict whether you can acquire those things. And although this is counterintuitive from one perspective, based on what you said before, if you think about it in evolutionary terms, from an infant, there's a 100% correspondence between the presence of the mother and the presence of the milk. So it sort of makes more sense that it would be sensitive to these predictive sensory cues.
Yeah, absolutely. I think that's correct. Uh, of course, there's some experiments that we haven't done and they should be done,
Mm-hmm.
and they're very difficult to do.
Yes, yes.
Hence why they haven't been done. And one alternative, so what you just said is the evolutionary explanation that we discussed before. So
Mm-hmm.
breast milk becomes irrelevant, the mother is a predictor, hence the proximal signal for the infant is the presence of the mother.
Mm-hmm.
The milk has no regulatory function. Another explanation will be what we would call a developmental explanation or an ontogenetic explanation.
Mm-hmm.
It will be somewhat like this. The infant, the AGP neurons actually are responding to deprivation of nutrients or let's say a metabolic challenge
Yep.
that's somewhat similar to food deprivation.
Mm-hmm.
And very early, the mother provides the milk, so fulfills that need and provides all of the sensory experience.
Mm-hmm.
The animal learns so rapidly, so strongly that from now on, it only use the proximal learning cues and doesn't use the nutritional cues anymore because they are irrelevant. So it will be that same evolutionary concept, but now placed in a developmental concept. So
Yep.
from birth to that point, so the animal learned that contingency, learned that pairing, and now is not using the breast milk as a cue.
Mm-hmm.
So we don't know that. We don't think that's the case because of all these studies that I told you, but it's very hard to prove that something doesn't happen or
Yeah,
something
yeah,
doesn't
yeah.
exist. And I think the experiment that will be important to do is to be able to raise these infants in the absence of a mother, for example, with an artificial us bottle feeding them from birth and then uncoupling maternal caretaking from ingestion and see what happens. Uh, the problems with that, those are very challenging experiments to do in animal models. Second, when you do that, you cause a lot of collateral damage
Yeah,
because you are
yeah.
you are you are breaking a contingency that's, as we said from
Mm-hmm.
the Harlow's work and others, is very important for development.
Yeah.
So but
I think another thing that's a little tricky here too is we're talking about rodents, so they're smaller creatures, and obviously baby rodents are even smaller than adult rodents, and their metabolism is very fast, and they're very sensitive to things like temperature. So I would imagine that, at least in principle, the brain of an infant's probably going to learn early on that when you're detached from the mother, obviously you're away from the source of milk, but you also immediately have considerations, for like hypothermia here, and so the brain could learn very quickly. When the temperature drops, I'm about to be nutrient deprived,
yeah,
because mom's not around or things like that.
yeah, the way we think is very similar to that, but it's broader in terms is not only nutrient deprivation, but as is a metabolic challenge in general.
Mm-hmm.
The infant, if it needs to produce more heat,
Right.
it will be a net deficit
Yep.
in
Yep.
energy. And originally, when we published that study in 2019, now 5 years ago, we did test that idea that temperature was an important component of that, and it is so. And how did we do that? Basically, remember, if we socially isolate the baby mouse, AGP neurons, the hunger neurons, quote unquote, are highly active. So what we did was to provide a warm environment.
Mm-hmm.
If you provide a warm environment, that's the temperature of the nest, then you prevent that activation or you block substantially that activation.
I see.
And we also did an experiment to test whether there was learn. In other words, did they learn that when they are alone, it's cold and that generates a temperature shock that needs to amplify metabolism. So it's a learn. So what we did was to put mothers at thermoneutral temperatures. Thermoneutral is when you don't need to produce heat from your body to warm yourself up.
Hmm.
So we raised mothers at 35 Celsius, so the temperature of the nest. They delivered the babies at that temperature. So the babies never saw anything lower than 35 Celsius, so they were always warm. They never had to produce heat.
Hmm.
And 10 days later, around the time that we did the other experiments, we separate them from the mother.
Yep, just like you did before, but now temperature is not a variable.
Yeah. So we separate at 35 Celsius, so they are just alone, or we separate them at a colder temperature, room temperature. So if we put these babies alone uh, at 35 Celsius, we have a very minimum activation of the neurons.
Mm.
Really, really, really minimal.
Hmm.
So almost no activation. If we put the babies at room temperature, we have an incredible activation.
Hmm.
So they don't need to learn the contingency.
Yeah, yeah.
It's something that the system is capable of responding without experience in that
Yeah.
regard.
And so, if I'm hearing you correctly, I think this is what you were getting at a moment ago. It's as if the neurons are anticipating metabolic need, and obviously, food is an important thing related to your metabolic needs of the body, but it's not the only thing.
Exactly. So temperature, other forms of, if for an infant, very likely temperature is one of the primary needs for the infant, given what you said, they have high metabolic rates. And even the work of Harlow, as I said before, I think speaks to that fact because when he cools down the mother, the babies will not attach to the mothers. So
Hmm.
temperatures seems to be an experience or heat from the mother, a sensory modality that's very important for
Hmm.
the infant. Yeah.
Hmm. And sort of, given everything we're talking about, how, hunger needs and developmental needs change at different phases of the lifespan, I would imagine that a lot of the circuitry changes or develops around a critical period, which I would guess for this would be around the time of weaning, because that's when the animal stops needing its mother for milk and can start eating solid foods. Do we know anything about if the circuitry is changing around that time or anything like that?
Yeah, we know a little bit about the circuitry change, but I would say definitely not enough. And we have follow-up work that's unpublished, but I'm happy to share that touch touch on this issue. So on the circuitry, we know actually that the HRP and POMC, this feeding satiety neurons, they have a lengthy development. So they would, in the mouse world, they will develop all the way up to 5, 6 weeks after birth, which means that's middle to late adolescence. So they will be increasing projections, axons going to places. They will have more synapses into postsynaptic areas. So that process is ongoing for all these weeks. Now, most of the places that will receive a projection, for example, from the AGP neurons, so a postsynaptic brain area, will already have a connection in the second week of life. But some of these connections will be very weak.
Hmm.
So they will be increasing in density as the animal ages and goes through adolescence. So we know that's so we know a little bit about that. Uh, so we also had that prediction that the weaning, when they are independent of the mother, so they don't need more the warmth or any of that, should be a moment in which this response to the social environment, one would say, would not be significant for the activity of these hunger neurons anymore, or these
Mhm.
metabolic neurons. So we have been working on that question since then for like 5 years now. And it's very surprising. So if we take an adolescent mice and now we socially isolate this mice, let's say a mouse that's in early adolescence, 25, 28 days old, haven't seen the mother for a week, is eating on their own. We separate these adolescent mice for a couple hours from their siblings. And when we separate them, we give them food. So if they want to eat, they can do whatever they want. We see the same activation of the AGP neurons that we saw in the babies.
Hmm.
So actually, the effect doesn't go away.
Hmm.
The effect only goes away in late adolescence
Hmm.
when they are in the last stage of maturity, sexual maturity, when they really become sexually mature, and potentially when there is a switch, we can talk more about the value of social behaviors.
Hmm.
So we still don't fully know the mechanism those findings are actually unpublished, and but it goes beyond weaning, which is quite striking actually.
Yeah, yeah. Another thing I'm interested in here is, so, I mean, as with any circuit, right, there's ways for hunger and satiety circuitry to develop in a healthy or normal or natural fashion. And then obviously things can go awry. And there's many ways for brain development to go awry. Some things that come to mind here that you might know something about, maybe you've done experiments on, are, I imagine there's a lot of individual variability, even in something like maternal care behavior. We see that in the natural world. I would imagine that some mouse mothers are very attentive mothers. They're always there. They're rarely separated from the pup. There's always there to allow the pup to suckle when it needs to. And maybe some other mothers, leave the nest more often. They're separated from the young more often. And there's also the question of, say, variability in the mother's diet, the composition of the diet, and how that affects the maturation of their pup's hunger circuitry. Do we know anything about how any of those variables can lead to, say, abnormal development in the hunger circuitry, how, maybe the absence of enough maternal care behavior or the absence of certain dietary components might lead the hunger circuitry to actually develop in ways that are, maladaptive, that leave the individual vulnerable to metabolic syndrome or something like that?
Yeah. Wow. Amazing questions, Nick. A lot to unpack there. So just to myself rephrase you now, try to understand if there's anything out there that we have done or others trying to understand variability in maternal effects and how that affects the development of these circuits.
Yes.
Uh, we have we have not done those experiments, but two uh, lines of research or findings come to my mind. All of them relate to nutrition and diet or availability of nutrients. So the first is to, as you said, to switch the diet of the mother.
Mm-hmm.
For example, in the mouse world, we switch to a diet rich in fat, and so a high fat diet. So the animals will put more weight and things like that. So if you do that during lactation in the mouse, that's the work of Jens Bruning and Max Planck in Germany. So the sensitive period seems to be lactation. So if the mother is eating a high fat diet, that impairs the development of the AGRP neurons.
Hmm.
So the impairment means there's less projections of these AGRP neurons to some key target sites or postsynaptic sites, and those infants that now grow, when he puts them on a high fat diet, they will become more obese. So it seems that the diet of the mother affects the development of these neurons and leaves a mark that will
Mm-hmm.
affect how the infants will respond.
I see.
Uh,
So if you give a mother a high fat diet during lactation, that's presumably affecting the composition of the milk and therefore what the infant is ingesting. When people do that experiment, it affects these AGRP neurons, the density of connections to other brain areas. And then if you let those baby mice grow up, they're then more prone to develop obesity if they have a high fat diet themselves.
exactly.
Hmm.
And a second uh, experiment that was done to cause overnutrition
Hmm.
in the mouse world. For example, a mouse mother will have on average 7 babies, 8 babies, 10 babies. So when the babies are born, if you cut the litter to like 3 babies or something very small, they will have a lot of milk available
Mm.
because now there's no competition. So in that way, you can have a model of overgrowth. They
Hmm.
will They will grow quite a bit and put a lot of weight. So there's a phenomenon that happens on the age of neurons. Gets a little complicated, but it's very elegant. Uh, so this is this hormone leptin produced by the fat discovered in 1994 by Jeff Friedman. Leptin. So when there's a lot of fat, leptin is high. Leptin suppresses the activity of the HAP neurons. So it should decrease this hunger state overall. That's a simplified conceptual framework.
Mm-hmm.
Now, the interesting thing is that leptin levels are very high in infants
Hmm.
in the first week, second week of life. And that leptin, during that period, that leptin actually doesn't inhibit the HAP neurons. The leptin activates the HAP neurons.
Hmm.
So it has an activatory effect. And that effect of leptin on the HAP neurons, activating the HAP neurons, is important for the axons, for the synapses, for the neurons to grow to postsynaptic targets. That was a very elegant work of Richard Simerly. So I'm saying that because leptin activates the neurons and then suddenly switch to be an inhibitory signal because the neurons start expressing a particular type of potassium channel. So there's a shift in the expression of
Yeah.
channels in the
So at some point in development, these neurons start expressing new channels, and something that used to activate them, leptin, a gut hormone, is going to come to inhibit them after they cross that key threshold.
Exactly. So the work of Laurie Zelter at Columbia, she showed that in a model that you can do this overgrowth, having like 3 babies being raised by the mother.
Mm-hmm.
So now they have an overnutrition. You can actually shift the point in development in which that particular channel start to express. So leptin start to be that inhibitory signal early on. So you can shift in a couple days in the mouse that. So I'm telling you that as there is plasticity, one would say, in the system is responding to the environment in many ways. We know very little still about it. But is that there is definitely a lot going on there that will leave marks in the way the circuits are working then and will work in the future.
Yeah. And so it's very easy to imagine that, if the mother's diet changes its composition, her leptin levels will be different. You could have something shift when this sensitivity to leptin switches in the baby. And all of these things will, in different ways, affect how these HRP neurons are growing and how they're hooked up to other brain areas. And then naturally, in the adult version of that animal, that's going to affect how they respond to food and their feeding behavior and all that.
Exactly. And so, yeah, that's where we are at. We don't know much of those regulatory mechanisms, but we have these examples that tell us that
Mm-hmm.
there's a lot going on there. Yeah.
Yeah. Do you have children of your own?
No.
No, but neither do I. But it's still nonetheless, I'm naturally thinking about a variety of things for humans right now. All this work that you've done to look at the maturation of the system, the relationship between the mothers and the infants, the importance of the different sensory stimuli and the whole context for the learning of the infant, and how all of these things regulate the development of the animal. What does that start to make you think about when you think about things like, human breastfeeding versus bottle feeding versus all of the sort of artificial things we invent for babies to give them, sometimes in place of what would otherwise happen naturally? Is that something you've thought about?
That's a very delicate topic, and I'm not a specialist to actually give a thoughtful answer to that. What I think and what we have learned, I think, from animal studies and from a very large body of human literature as well, is that the stimulation of the infant, the caretaking, that's very important for the neurodevelopment.
Mm-hmm.
So even in the absence of natural breast milk, for instance, it's the infant can develop normally by receiving all the stimulations they need. It seems to me that the most critical part is to deprive the infants from the proper stimulations that they need.
Hmm.
Uh, there is uh, there's an example that I like a lot. Let me try to remember the details, but it's fascinating because during World War II, uh, one of the last places for to achieve peace was Okinawa Shima in
Mm.
Japan. So when the American army was there, the island was obviously devastated, uh, and they noticed something that was striking to them. To remember, that's 1940s, before Harlow experiments and all of that, all of
Hmm.
the above. Uh, they noticed that despite the devastation, there were no psychiatrists in the island. There were no such a thing as a psychiatric hospital, and people were really mentally stable.
Hmm.
Uh, they just came from a war. Many people were injured, and there was not much going on in terms of psychological
Yeah.
issues.
They just went through this major trauma, but no, it wasn't like everyone was walking around with severe PTSD.
And back in the day, half of the beds in American hospitals were for mental health.
Hmm.
So mental health had a huge cost. So they were really puzzled by that. And so they made a movie. They filmed Okinawa Shima. And the movie is called The Okinawans.
Hmm.
And in this movie, they show all this data and they say there's only one thing that we have observed that we can think that is what's causing this psychological stability or strength of the Okinawans. And it doesn't seem to be a change in diet, a change in genetics, or anything like that that compares to folks in the Western world. But it seems to be the way that the Okinawan newborns are mothered.
Hmm.
So for the first 2 years of life, the Okinawans are fully under uh, caretaking of their mother, 100%. So every sign, the mother will pay attention, will take care of what the baby is in need, and often will give the breast for both nutrition or not
Hmm.
as a calming effect. From 2 years old, the oldest daughter, if there's no, if the family has, the baby doesn't have a oldest daughter, it will be somebody else, cousins or other
Hmm.
siblings that will take care of that infant from 2 to 5 years.
Hmm. So
Which
there's always someone around in the nest.
there's always somebody around in the nest. That baby will never be in the playground alone.
Hmm.
And then say the only explanation that we can find for this psychological strength of the Okinawans is the fact that they have these strong relationships and they don't have any distress or adversity in their life, so they can control their emotions fully.
Hmm.
And that was a really remarkable finding. It's a beautiful movie. It's and which anticipates all the experiments of Harlow and attachment theory and things that we are talking until now. So I think that kind of findings that we know a lot now experimentally is that this sensory, the caretaking from both parents. I talk a lot about mother, but doesn't need to come from the mother. Can come from the father. Can come from uh, um, other caretakers. It That's what's important for the developing infant to have that sensory exposure.
Hmm. Hmm. Wow. So yeah, always having someone in the nest.
Yeah.
Interesting. Interesting. Um, Another thing I want to talk about that it sort of relates to this, but we're going into a bit of a different topic now. You study individual variability in behavior and how that develops. And I haven't looked into this in too much detail, but I know that's a topic you study, and I know that you also seem to be studying it using a very peculiar animal. Can you talk a little bit about what you're doing there?
Yeah, it's a little bit of a changing topic, but I have always been fascinated by this concept of individuality and how we come about to be what we are. And and of course, there's a lot of debate out there about the nurture nature, uh, um, the importance of nature nurture for development, and then for individuality, which means the genetic makeup of an animal and all the environmental experiences that they have. And I'm particularly interested in these concepts in mammals that have a complex brain, have this postnatal period that they have a dependency on a caretaker. In that period, the brain is growing, the cortex is growing, and the baby is learning all these contingencies and being exposed to all these sensory experiences, which is quite unique to mammals. So I wanted to try to study that in a model that I can control the genetics and that I can control the environment. Uh, in other words, if I would have four nicks in front of me, four monozygotic twins, clones that were raised in the same conditions, would the four nicks be interviewing me for the podcast, or maybe one will be sitting on my seat, or what would happen?
Hmm.
And would they have the same diseases or not? And I think that's important because when we look at the data of, for example, brain diseases, and we look at the human data of monozygotic twins, so twins that are genetically identical, usually we don't have 100% concordance. When is a disease that have high concordance, which means if one sibling has the disease, the other will have.
Hmm.
In brain diseases, the maximum you can get is really 80%, but it's typically below 40% of concordance.
Hmm.
There's a lot going on in the way the brain develops and the way individuality develops. So, how to study a complex mammal, control for genetics, and control for the environment. So, when you look at the mammalian tree of life on the 6,500 mammals, there is one mammal that I grew up with them in Brazil. That are the armadillos. So the armadillos are the only mammals, one particular species of armadillos that always give birth to four
Ah.
clones, always give birth to quadruplets. So it's such a fascinating experiment of evolution to create this striking animal, has a brain, has a complex brain, it's a mammal, needs a caretaker, and always generates four identical
Hmm.
siblings.
So you've got identical genetics, as identical an environment as you're going to get, but nonetheless, you've got now four separate individuals.
So you can have statistical power to look for how different they are or how similar they are. So that's one line of research that we are trying to do in the lab. I have very little to talk about it besides this conceptual framework. We are still, uh, I would say, we are
Developing the system.
developing the system is very tricky. I have been working on that for like a decade now, and now we are trying to generate a colony. We have 14 animals in my lab,
Hmm.
so I think we are close to a point, an inflection point that we will start having more to say.
Hmm. But in essence, what you're going to be studying is the neural basis of personality.
Yeah, exactly, one could say it that way.
Yeah.
Yeah. Uh,
And how much, can you give us a sense for, how much natural variation do we see in laboratory animals like, the inbred strains of mice that people normally use? Is there a fair amount of variability there?
yeah, that's a
Kind of
yeah,
hard to say, maybe.
there's a huge amount of variability.
Mm.
And so yeah, I'm so mouse models, rat models, back in the day, they were thought that the best way to create these models as an experimental model system is to control their genetics. So you
Hmm.
do sibling to sibling breeding. You breed them, breed them, breed them, breed them, breed them, breed them, breed them. So you create this inbreeding to a point that they are, I would say, quasi genetically identical. So
Hmm.
all the genes are the same, and they are all two copies of the same. So they are all homozygote for those genes. So those are what we call the inbred mouse lines that we use. The
Hmm.
most classic used in neuroscience, C57 black 6. So which means that animal has been bred for now 100 plus years in controlled conditions, and they are fully homozygote. When you try to do that kind of experiment, actually, that kind of inbreeding, actually a minority of these inbreeding conditions will actually generate a line of animals that you can in fact inbreed. Many of them become infertile because
Yeah.
this the homozygotic uh, being fully homozygote is really not a natural thing.
Yeah. Yeah.
So, and the animals are never 100% identical because there's always what we call a little bit of a drift in the
Yeah.
order of
Yeah.
1%.
But
But
they're much more similar than
yes,
two random people on the street would be.
they are much more similar than two random people on the street. So the prediction was if you do that in breeding, you have more constant genetic, you decrease variability
Yeah.
in traits in general. And
Including
that was
behavior.
behavior and any trait,
Yeah. Yeah.
size, body size, organ size. And surprising, that's not the case. You still have variability even in these inbred mouse lines, which is equally to the variability that you find, for example, if you take two strains and generates what we call an F1. So
Hmm.
now it's a heterozygote from two strains, or if you take outbred strains, which are strains that are not genetically inbred.
Hmm.
When you look at the variability, for example, the percent that that trait varies compared to the mean of that population, actually inbreeding does not decrease variability.
Huh. Wow.
Yeah. So there's a lot to talk about it, but to me, one of the most remarkable experiments was done in Germany over a period of about 30 years, studying traits of mice and showing that this variability persists despite being breeding. And what was called back in the day, 40 years ago, was that's a third component. What is the third component? It's not the genetically constant. You have animals raised in semi-identical environment, and the variability always remains. There's a chunk of
Hmm.
variability that you can never suppress.
Hmm.
That's a third
Yeah.
component.
I mean,
And what is
to me, what
yeah,
I think of here before I let you take it away, but I had a conversation with a neurogeneticist named Kevin Mitchell at one point, and one of the things we talked about is just I don't know what the official term is, but it's developmental stochasticity.
yeah,
There's just sort of a natural randomness in how many of the biological processes inside of us happen, such that even with identical genetics and identical cells, otherwise, sometimes things go in this direction and sometimes they go in that direction.
exactly. Yeah, Kevin is one of the, I would say, big thinkers on this field. That the stochastic developmental variability, the higher the complexity of the system, you have so many steps, you introduce some kind of random process and you create a cascade
Mm-hmm.
that propagates. So you end up always with systems that are different. And that is certainly very important for the nervous system of a complex animal like mammals. So yeah, this is called SDV, so the stochastic developmental variability being the third component. So this component that cannot be controlled by genetics
Mm-hmm.
inbreeding and cannot be controlled by environmental constancy or decreasing
Mm-hmm.
environmental variability.
And if we sort of put those things together, so you told us that we've created all these inbred mouse strains, but they show comparable levels of variability across traits to non-inbred strains. And so this would say that the developmental stochasticity is a very significant component of the variation that we see.
is a significant component and is a critical component. And it's part also tells us that's part of the adaptive value of the evolutionary context. We evolved, animals evolved to have that component as part of the variance, as part of the variability. As we know in evolutionary theory, you can only select for things that have variance.
Hmm.
You need to you need to have variance to be able to select for certain traits. So it's striking to realize that actually there is a selection for the presence of variance as a default on the top of genetically
Yeah.
identity environmental.
There's always variation for natural selection to work with, even in the absence of genetic variability.
Exactly. Now, why would that be the case? Right? Because that's not being, for example, that if you generate four Marcellos and they are all genetically identical, the only tool the natural selection e- evolution has to work is on my DNA. What
Yes.
is best?
Yep.
The structure of my brain is not going to necessarily pass to the
Yeah.
offspring.
Yeah. I mean, the difficulty here conceptually for me is, if we have four Marcellos and they have some variation that's completely due to this developmental stochasticity thing, let's say two of them are adapted to their environment and two of them are not, they die off. Well, if the two of them that are adapted to the environment such that they live long enough to reproduce, their babies aren't now they're not necessarily going to actually inherit the traits that were adaptive for you because they're actually not encoded in the genes.
Exactly. So that's actually a puzzle. I don't think we have
Mm-hmm.
a good solution for that. And it's a puzzle in evolutionary theory why, for example, we have armadillos that give four offspring. Uh, because why not then generating four genetically different that at least you can select for those traits? And yeah, I don't think we have a solution to that discussion. Uh, one theory that in species that might have high rates of predation, so generating clones, you might have a higher chance of passing. So you generate clones, you generate variation in behavior, they spread out in the environment. In the case of armadillos, they are solitary, so they can occupy a larger territory.
Mm-hmm.
They have high rate of predation, so the chance of one surviving
It's quite
is
low.
is quite low. So you are actually never or very low probability having two of them surviving and their offspring. So basically, you are multiplying the self. Increasing the capacity of spreading your genes.
Yeah,
But
yeah.
so that's one of the I think the most accepted ideas behind that. Uh,
Interesting.
yeah.
And then, so what are some of the things that you guys are working on right now in the lab, especially on the maternal care side? And in the first part of our discussion, what are some of the upcoming questions you're trying to tackle, and where are things going to be going in terms of what comes out of your lab in the next couple of years?
Yeah, we talk about this armadillo world. I think this is going to be beyond couple years, and we talk a little bit about this unpublished work on adolescence and this realization that these feeding or hunger neurons are still being recruited for social behaviors during those sensitive periods of adolescence. So we, a part of my lab is heavily in, invested in trying to understand at the circuit level what's happening there. So when the infant or an adolescent is alone, how is that information coming to the aged neurons to activate them? And are those circuits different than the circuits that are involved in the response to food or
Mm-hmm.
in the response to temperature? So can we understand more about that? And would that give us insight of what's actually happening there? So part of my lab is trying to understand that. And another big part of my lab is trying to understand how does the infant make this concept of a caretaker?
Mm.
So how is a caretaker, one would say, encoded at the level of the brain? Where is that information? And and then how does that information is used for future social behaviors?
Hmm.
In other words, this primary social relationship of the infant with the caretaker is that is that relationship at the neurocircuit level being useful, and how so for future social relationships that have distinct characteristics. An adolescent that's playing is a very different social behavior compared to an infant that's calling attention of a caretaker. And an adult that's engaged in sexual behavior, or territorial behavior, or other kind of behavior is still another kind of social relationship. Are the social behaviors of different periods of life somewhat related to those early relationships with the caretaker?
Yeah.
And and how
Yeah.
so? So we are really going to that direction at the neurocircuit, neurocoding level.
Yeah, yeah. I mean, it's very natural to think that there's going to be significant findings there because, I'm thinking of the Okinawa movie you told us about. Someone, if you're growing up in a situation where someone's always in the nest, you always have a loving caretaker, you're probably going to have a very different propensity to develop certain behavioral tendencies, certain disease tendencies or disorder tendencies compared to if you grow up in an environment where people are absent more often or someone aggressive is around, et cetera, et cetera.
Absolutely. So there's at least two ways to broadly think about this. One is that the relationship, the calming effects of the healthy relationship affects in some ways how other systems in the body develop, and hence the long-term effect of that relationship is by leaving these marks.
Mhm.
Okay. And the other way to think is that at the neuroencoding, for example, their relationship with the mother is encoded as somehow in an emotional sense, in a neurocoding sense. And then later in life, when the animal is subject to some events in life, you will use that information
As a template.
as a template.
Yeah.
And I think that's important for social behaviors.
Yeah. It reminds me a lot about the basic way that people who study songbirds study vocal learning. There's a critical period in which a template develops and crystallizes. Sort of every subsequent learning event is in many ways compared to that initial template.
Yeah, absolutely. There are some experiments that are quite mind-blowing that were done in the late 60s, second half of 60s, 1960s. Basically, they raised mice with rats. So they took mouse babies and they gave to rat mothers. And those mouse babies raised by rat mothers, when they become adults later in life, they will choose to socialize with rats, not with mice. So the idea being that there are inborn tendencies for social animals to socialize.
Yeah.
However, the experience of Joaquin of what a social object is actually learned
Yeah. Yeah.
is learned during
Yeah.
sensitive periods of life, and then
Yeah.
in the mouse seems to be during lactation,
Yeah.
and
Yeah.
so they
And
come.
there's so many fun YouTube videos out there where people say things like my cat thinks it's a dog or things like that. You see the cat
Exactly.
running around with the dogs instead of the cats or whatever.
Yeah. So, anyways, those are kind of things that we are trying to study at the neurocircuit base and the
Hmm.
neuroencoding base.
Yeah. Well, this has been fascinating. I really like the spectrum of work that your lab does. It's really cool stuff. Are there any final thoughts you want to leave people with? Anything you want to reiterate, either about your scientific work and the things that we talked about, or maybe even how your work has influenced how you think about social relationships and how we relate to each other as humans?
Yeah. We'll start with the sentence of Harlow that we have to learn how to love before we learn how to live. And I think, Yeah, I have had my lab since 2014, so 10 years now, and I think working on this field and doing some of these experiments that we do really brought that concept close to heart. And I think, if we look at things, for example, the NIH budget, which is the largest entity of funding in the world, I think the NIH budget to Alzheimer's disease year tag, if such a fundamental problem, is about $6 billion a year tagged. I believe the NIH budget for the National Institute of Child Development, which means the entire research that the country does in child health and child development, including clinical trials, everything included, I think is under $2 billion. So I think if I would leave a message is that development is very critical for health in general. You understand an infant is not a mini adult. An infant is a complex dynamic system that is solving physiological problems then and anticipating for the future ones. And this machine is beautifully working and orchestrated. And whatever we do, as you said, change diet or early life stress, or it will change how the systems develop. But that's a very fundamental problem to work. And it would be amazing to have more people joining the forces and thinking about these problems together is a fascinating moment of science. I tell always my group, it's just mind blowing the tools that we have and the depth of the questions that we can ask. It would be amazing to have a vibrant community in that regard.
All right. Well, Professor Marcelo Dietrich, thank you very much for your time. Fascinating stuff. And I hope to talk to you again at some point.
Thank you, Nick. It was a pleasure.